Welcome everyone. It's great to see many of you here again. So today we have Dr. Anju Gupta. She is an assistant professor in the Mechanical, Industrial and Manufacturing Engineering department at the University of Toledo. And her area is thermal fluid science, materials design, manufacturing, and today you will hear her work, and she has been recognized from ACS petroleum research funding for new faculty, and her work has been supported by NSF, and her graduate students are also have received many prestigious awards, including the Ohio Space Grant and university fellowships. So she has co-authored more than 35 peer-reviewed scientific and educational journals, and with many featured journal articles. And so she has had many collaborations from NIH and Oak Ridge National Labs. And so I think today you will hear her work on phase change in lipid bilayer. So with that, I would like to give the stage to Dr. Gupta.
Thanks for the introduction, Yang Ying. I also want to thank Matteo and Mohammed for the opportunity to share my work with thermal transport cafe community. And thanks for the patience. I know I was scheduled to give this talk last December, but somehow things didn't work out. So I'm happy to present my work here today with you. I want to begin by recognizing my wonderful graduate and undergraduate students, Ali, Saket, Mohd, and Turkey. It's all their hard work that we are able to get financial support received from various federal agencies and industries. Since I'm fairly new to this community, I want to use this platform to introduce you to various interfacial thermal and transport projects in my lab that we apply to energy, environment, and medicine. Some of you are familiar with our recent work on magnetically aligned copper and nickel links for pool boiling applications. So we created grids and channels like microstructures that yielded very high critical heat flux at low wall superheat compared to plain copper. We attributed this performance to microlayer evaporation and enhanced three-phase contact angle. We are also creating mixed matrix membranes by combining hydrophobic polymers with carbon fibers for desalination application. So here we have our seawater or produced water from a petroleum or water that has rare earth metals. We heat it up and the vapors pass through these hydrophobic membranes, and then we condense down the purified liquid. With this membrane, we are able to achieve high water vapor flux compared to commercially available membranes, and a very high salt rejection rate of around 99.99%. In this talk, I will talk about thermal analysis of lipid bilayer. It is still thermal and transport, but it's a little different than what we see in this cafe talks. I hope you find this work interesting. So my big application is photodynamic therapy. And in this talk, I will show you how, as a thermal scientist, we can contribute in increasing the efficiency of photodynamic therapy. This was a problem that we accidentally stumbled upon while we were interacting with laser surgery department in Rochester General Hospital. So I will begin my talk. But first, I want to give you an overview on why we need to study lipid bilayers and why it's an interesting problem and it's a pressing issue. So cells, as we all know, are the basic units of life. They are protected by plasma membrane. And the plasma membrane is responsible for allowing the passage of materials in and out of the cell. The cell membranes have to be in fluid state or have to be permeable for their normal cell function. And the interaction of cell membrane with potential drug candidates is an important step for drug discovery process. Unfortunately, the mechanisms by which the drugs bind or interact or translocate through cell membranes are still unknown, but we are all trying our best to understand this mystery. Cell membranes are primarily made of lipids, along with some cholesterol and membrane proteins. And lipid by themselves or lipid bilayer themselves serve as an excellent model for cell membranes, because as we all know, it's really hard to study cells because the cell membranes are very complex and tiny. So lipid bilayers are an excellent model to look at the interactions of drug and potential therapeutics with cell membranes. Lipids are very similar to polymers, because if you heat them up, they undergo temperature transitions. You see them going from gel to fluid phase, which is what I'm going to talk about in this work. And so if we are able to understand how potential drugs or therapeutics bind with lipid bilayer and translocate through lipid bilayer, and while doing so, how it's creating thermal disordering and partitioning within the bilayer, we can definitely solve the mystery of how drugs interact or internalize in the cells. So now the challenge remains twofold. How do we look at the structure of lipid bilayer, and how do we study these thermal disordering within through the partitioning? And we want to do that without using external probe molecules. So in my research lab, we have been using a very simple technique, which you all are familiar with, differential scanning calorimetry. And we use this equipment as a rapid and inexpensive tool to study the thermotropic or fluid behavior of lipid bilayer. And then we feed that experimental data in our computational simulations to see what's happening to the drug or the lipid bilayer. And in the very recent times, we are trying to feed all that information in our machine learning model. And I'm going to show you where there's a lot of opportunity for machine learning experts to collaborate with us. So in this talk, I will begin by introducing you to phospholipids, how they look like, how they self-assemble and different shapes they self-assemble in, and how we characterize them. I also want to focus on liposomes and their applications in medicine, because if you're looking at thermal analysis of lipids, you're solving a lot of problems in biomedicine. And we have been focusing mostly on self-assembled lipids in bilayer vesicles, which are also called liposomes. So that will be the flavor of this talk. In the second section, I'm going to show you, or I'm going to focus on differential scanning calorimetry thermographs of lipids and how that changes when you have cholesterol or polymers embedded in the liposome or on top of liposome. And with that background, I'm going to introduce you to, or maybe reintroduce you to, photodynamic therapy. And we recently learned how lipids, heat transfer, transport phenomena can increase the efficiency of photodynamic therapy. We have been working on this for quite some time. And in the interest of time, I'm only going to focus on very small section of our work on photosensitizers. So we discovered / modified existing photosensitizers, and we kind of predicted how they are going to perform if they were used in photodynamic therapy. So I'm going to talk about just two types of photosensitizers. We did some computational modeling. If the video works, I'll be happy to show you how they go in the lipid bilayer. And that would be all for today's talk. So there's a lot to unpack, and let's get started. So this is a schematic of a cell and its organelles. You will notice this cell and all the organelles are protected by membranes. And these bar graphs surrounding the cell shows you the composition of lipids, different lipids, some cholesterol, and other biological materials that look similar to lipids. Let's take a look at plasma membrane. So plasma membrane, based on this bar graph, comprises of PC. PC is phosphatidylcholine lipid. And if you look at the bar charts throughout this slide, you will see PC is present in abundance in almost all the membranes. So phosphatidylcholine or PC lipids are present in both eukaryotic and mammalian cells, and they are zwitterion. It's very important to understand the charges of the lipids, and I'll show you why in next few slides. So we call this a zwitterionic lipid because it has a positive charge and a negative charge moiety, but together there's a net charge of 0. And the second type of lipid I want you to focus on is PS, which is also present in most membranes, according to this figure. Phosphatidylserine is, again, a negatively charged lipid. The reason being there are positive charges and there are negative charges, but in physiological pH, it gives a net charge of -1. And that's because the pKa values of carboxyl group here is very low compared to the pKa value of ammonia, which is around 9.2. So the net charge comes down to -1 in physiological pH, and which is what we use in most of our studies. We create our systems to have a physiological pH environment. So in all my work, I will be using PC and PS lipids. So starting now, all the work that I'm going to show you, if I forget to mention what composition of lipids I'm talking about, it's mostly going to be PC and PS. I also want to point out that the biological membranes have a slight negative charge, and therefore all the prodrugs, peptides, and proteins we design are typically cationic in nature, or they are mostly positively charged, so they are able to electrostatically bind with the biological membranes. So lipids look very similar to surfactants. They have a hydrophilic head group and a hydrophobic tail. They can have single tail, they can have double tails. The tails can be saturated or unsaturated. In an aqueous medium, these lipids can self-assemble to either form micelles or bilayer structures like this, or inverted micelles. So for all our work, we create bilayer vesicles, which are also known as liposomes. So this is a schematic of a liposome. It has an aqueous core, which is hydrophilic, surrounded by hydrophobic bilayer. So this resembles a cell, and therefore liposomes can be an excellent substitute for cells. And if you're trying to play around with cells or trying to study the interaction, liposomes serve as an excellent model. We create these liposomes by self-assembling lipids, in a variety of biological fluids, PBS, DMSO, sometimes just deionized water. This figure is a cell phone photo of a liposomal solution. So I used PC lipids. We suspended them in PBS buffer, and you see different cloudiness, which represents different concentrations of liposomal solutions. And on the right is the cryogenic TEM image of these liposomal solutions. So if you are running electron microscope in cryogenic conditions, you are able to view the colloidal particles. You don't have to dry them out. Another interesting and versatile thing about liposomes is you can custom design them. You can pick and choose the lipids. You can also pick and choose the head group charges. If you want your liposome to be very negative, you can go for lipids that have negatively charged head groups. You can also size them. For most biological work, especially for drug delivery applications, we try to have liposomes of 100 nanometers in size, and we typically use polycarbonate membranes to extrude them. This is a raw figure or raw data from NanoZeta that gives you the average diameter of liposomes or colloidal particles, along with the polydispersity index, and it also gives you the quality of result. The x-axis is the hydrodynamic diameter in nanometers, and these micelles are seen around 10 nanometers, and our sized liposomes are around 150 nanometers. And in the field of liposomes, if your PDI is less than 0.4, it implies that you have a uniformly sized liposome sample. In the bottom is the raw data of zeta potential. The x-axis is the apparent zeta potential. So you see a peak around 0, and that's because this sample is DPPC liposomes, and I mentioned DPPC are citrinic. They have a net neutral charge, and so you end up seeing a single peak around 0. Again, in field of colloids and liposomes, if you have a zeta potential in the range of -30 to +30, you have a very stable colloidal formulation that looks like this, where you don't see any aggregates, and you see a uniform cloudy suspension like this. Liposomes are versatile, and that's the reason we have spent a couple of years studying them. They are used in a variety of medical applications. I also read an article some time ago. They were trying to use liposomes for soil remediation, which could be a very expensive proposition. Your COVID-19 vaccine is also lipid-based, and since it's able to encapsulate a lot of mRNA, it has gained a lot of attention. I would say in the last 2 to 3 years, we have seen a lot of literature on liposomes. So it's a great problem to solve at the right time. The advantage of using liposome is it has an aqueous core. And so you can encapsulate this with hydrophilic drugs or DNA, mRNA, or quantum dots. It also has a hydrophobic region, so you can encapsulate hydrophobic drugs which have ring structures that are more lipophilic in nature. And once you create these cargos, you can use them for diagnosis of diseases. You can use them for vaccine therapy, and you can also use them to kill cells in photodynamic therapy. In my lab, we have worked on several of these applications. We have combined diagnosis and photodynamic therapy together, which will be discussed in the second section of this talk. So in the beginning, I mentioned the reason we want to study cell membranes through lipid bilayer models is because we don't know the mechanisms, how drugs bind or interact. So if you look at prescription drugs, sometimes they write, although the mechanism is unknown, we see great clinical trial results. And this is the reason there's a lot of room in lipid bilayer literature to look at the interactions. However, there are 3 classical mechanisms that have been proposed in the field of cell membrane disruption. So cancer literature has made a lot of progress, and a lot of anti-cancer peptides and proteins are able to disrupt membranes through 3 major yet related mechanisms. So the first one is pore formation. You have your peptides that bind on the lipid surface, and they insert deep into the bilayer or the plasma membrane. They form channels or pores, and they disintegrate the membrane. In the second mechanism, these peptides aggregate on surface of the lipid bilayer, and they look like carpets on a large floor, and subsequently they start forming micelles with the lipids, and the entire lipid bilayer disintegrates. And we have seen these under cryo-TEM, and they look like discs as opposed to round liposomes, and that's when we know that the membranes have been disrupted and the anti-cancer drugs or peptides are working. Membrane disruption is also desirable in antimicrobial peptide field, where you're trying to kill bacteria, and the easiest way to do it is to attack their membrane. So there's a lot of interesting things going on in the membrane science. So I hope I have generated enough enthusiasm in you about lipid bilayer, and you are excited to see how you can contribute in the field through your expertise in thermal sciences. So in this section of this talk, I am going to focus on DSC of lipids. I will show you when you have cholesterol or polymers in your lipid bilayer or membranes, how they impact the thermal analysis studies, and how all that is relevant to photodynamic therapy. And again, I'll briefly show you some of our interesting results with newly discovered photosensitizers. So DSC, as we all know, is a widely used technique in polymer industry. So here you heat up a sample and compare that with the reference material. You can heat up the sample in a set temperature range in an inert atmosphere. And this is the typical data you get. So the y-axis typically gives you heat flow or heat capacity. If you can change the y-axis and add molecular weight, you can change the y-axis. On x-axis, it gives you the temperature. And this is an example of DSC of polymers. It's from my advanced materials course. Here you see glass transitions, crystallization, and melting peaks that are specific to polymers. This is a melting peak, which we also see in DSC analysis of lipids. So your S and L correspond to solid and liquid temperatures. This is melting temperature. The area under the curve is the enthalpy of transition from solid to liquid phase. And in our case, it will be from gel to fluid, which I'll show you in the next slide. So once you have your melting temperature and your peak, you can compute entropy of this transition. And from that, you can also compute Vent-Hoff enthalpy. So the Vent-Hoff enthalpy involves melting temperature and half peak width. I'm not going to focus too much on Wen-Thoff enthalpy, but in the recent times, we have used Wen-Thoff enthalpy to calculate cooperativity. So the cooperativity gives you the cooperative units. What that means is it can tell you how many molecules, maybe of a drug or an antimicrobial peptides, are getting associated with the lipids. So just by doing a simple DSE run, we can figure out what's happening in the lipid bilayer, how are molecules interacting. So in this talk, I'm not going to, I won't have the time to talk about our cooperative units or cooperativity analysis, but do know the location of the peak, the size of the peak, the shape of the peak gives us a lot more information other than just melting temperature. So as I mentioned earlier, again, cell membranes are composed of a variety of lipids, but to keep things simpler, to create simple models for cell membranes, in all my work, we use two kinds of lipids. So our bilayers are now mixed lipid bilayer. We use 𝗕𝗵𝗃, which I have shown you has a 𝗟𝗶𝗽𝗃 charge. That means it has a net neutral charge of 0 millivolts. And the melting temperature of 𝗕𝗵𝗃 is 42 degrees Celsius. So this is 𝗟𝗃𝗃, which is present in abundance in all membranes. And so it makes sense to have a PC lipid in your model. And we also include 𝗕𝗵𝗃s. 𝗕𝗵𝗃s has a slightly higher melting temperature of 55 degrees Celsius. It has net negative charge. So combining 𝗕𝗵𝗃s and 𝗕𝗵𝗃c makes for a very good representative model for cell membrane for our work. So this is thermograph of 𝗕𝗵𝗃c lipids, which is this, and it was generated on nano DSC. Here you see two peaks. The first peak is pre-transition, in which your lipids are rigid. Your lipid tails are rigid. They are like sticks. And if you heat them further, you see a very sharp melting peak. And in this range, your tails, the hydrophobic tails, are permeable and they are fluid. So if your cell membranes need to be in fluid state for it to function normally, because that will involve passage of materials in and out of the cell. This is a raw data of nano DSC, and this shows the thermograph of 𝗕𝗵𝗃c and 𝗕𝗵𝗃s combined lipids. If you compare the two plots, here you see single sharp peak because you have a uniform composition. It's all 𝗕𝗵𝗃c lipids, so they are all fluidizing at the same temperature. In this case, you see a wider peak and you see a shoulder here, and that happens because of phase separation. And if you deconvolute this peak, you will see 𝗕𝗵𝗃c rich domains, 𝗕𝗵𝗃s rich domains, and then somewhere in center you will have mixed domains, melting domains of 𝗕𝗵𝗃c and 𝗕𝗵𝗃s. So we have done a lot of deconvolution studies to really see what's happening inside of this broader peak. I wanted to show this to you so you compare. You have a comparison of DSC versus nano DSC. I'm not a spokesperson for nano DSC, but I just wanted to show you that these two are the same exact samples, and this was generated on regular DSC, and same composition, same concentration of liposomes. However, in DSC, we did not see a pre-transition peak, and the melting peak was broad. It was not very pronounced. And that is the reason we prefer to use nano DSC for lipid bilayer research because it's very sensitive and it picks up every small phase separation that can give more information on what kind of mixed domains you're working with. So that's the reason we prefer using nano DSC in our lipid bilayer research. So in this paper, I used both of these, and we were able to show how nano DSC fare better than regular DSC. So before I jump on to photodynamic therapy, I want to remind you that liposomes are versatile. They are used in drug delivery vehicles. They are used as cargos. And so your goal is to keep them stabilized and prevent them from aggregating. So polymers have been added to liposomes to increase their stability. In fact, your COVID-19 vaccine also has polymers along with the lipids. And so polyethylene glycol is commonly used and FDA-approved polymer. So you can buy lipids that already has polymers or PEG attached on top of it, and then you can self-assemble to form liposomes like this. Depending on the size and concentration of PEG, they can either form mushroom or brush-like structures. So this is an example where the schematic shows that the low-density PEG polymers form mushroom-like structures on top of liposomes, and the high-density PEG polymers form brush-like structures on the surface. In this review article, we talked about various polymers-based formulations and how adding polymers to liposomes can increase their circulation times because they don't get attacked by macrophages as quickly. And I'm not going to discuss too much about how what these peaks mean, but I just wanted to show you real quickly that depending on the molecular weight of PEG, we can see how it changes the lipid bilayer phase behavior on DSC. So this was the work we did with National Cancer Institute. So we created these liposomes and we, of course, encapsulated them with a drug. So these were PC lipids, and they had PEG molecules of 1,000, 2,000, and 5,000 molecular weight. And we were surprised that PEG 2000 was the most stable formulation in our mice studies. And if you just look at quickly the thermal analysis of these three formulations, you'll see that lower molecular weight PEG shows a very nice sharp peak, and that broadens with addition of higher molecular PEG molecules. So we did an extensive study, which is published in this paper. And in my first few slides, I showed you that along with lipids, cholesterol is also present in cell membranes, and cholesterol has been historically added to make the liposomes very stable. Or if you want to make liposomes as a model cells, you absolutely have to add cholesterol to see the impact of cholesterol. So this is a nice review article that talks about various properties that change if you add cholesterol. But for our study, we demonstrated that, hey, cholesterol is hydrophobic. It is going to accumulate between the lipids. And when that happens, it's going to change the packing of lipids. And we saw those effects even with a simple DSC. And so this was published back in 2017, where we created formulations with DPPC and cholesterol. This is just a regular DPPC, but when you add cholesterol, the melting peak shifts towards left. And we think that's because now the cholesterol is changing the packing and it's creating fluidization at a lower temperature. And however, some literatures also demonstrate that it decreases the permeation because now it's causing an obstacle in the fluidity. So in this paper, we created formulations with varying ratios of DPPC and cholesterol. And so some of this is true because if you add a lot more cholesterol, then you are actually adding to the rigidity and the melting peak shifts on the right. So again, there's a lot of mystery behind how lipid bilayer will behave when you have a foreign molecule stuck in the bilayer. And this is what happens in drug discovery process. We try to design drugs based on, as long as it can enter the cell by crossing the plasma membrane, it's going to be great. So in the last few slides, I presented some of our work with liposomal formulation, how we created liposomes, how we characterize them, and how things change when you add polymers and cholesterol to increase their stability, and how the sizes of cholesterol and polymer and concentrations will also impact the melting behavior of lipids. I'm going to switch gears here and I will show you the significance of thermal analysis of lipid bilayer in cancer applications, and also for larger photodynamic therapy, which can also include treating skin conditions. So PDT, or photodynamic therapy, uses photosensitizers. These are fluorescent or dye-like molecules. They are sensitive to light, and they are introduced into tissues and they are exposed to light of certain wavelength. It then forms reactive oxidative species with the oxygen that's already present in cells. And these ROS, or reactive oxygen species, kills the cells. We got introduced to PDT a few years ago by our laser surgery department. So we were making liposomal formulations for them. We didn't know what we were doing, what they were doing with our formulations, but they said, hey, these are the lipids that we can use, and this is the drug. Can you form very nice, stable colloidal solutions? And as a chemical engineer, you are always excited to form stable solutions because it requires a lot of labor of love. So we were creating those, and then we finally asked them, what are you doing with those formulations? And then they explained this PDT. And so I thought of concerts, right? When you have those bright concert lights, things get really warm in the room or the concert arena. And so I asked them, do you think this light or the laser might be causing localized heating? And they said, yes, we tried measuring, and then they did measure, and it turns out, at least the type of laser they are using, it turns out the localized heating was measured at a temperature of 42 degrees Celsius. So we thought, hey, if I have to make liposomes to carry anti-cancer drugs, and if I can make them with lipids that fluidize at 42, won't they have higher permeability, and won't they release more photosensitizers in the cells? And since then, we have been focused on the localized heating aspect of photodynamic therapy. And this was an accidental discovery, just because as a thermal scientist, we think about, is something getting hot? Of course there is. And since I'm teaching heat transfer, any temperature difference is very exciting for us. So in this slide, I'm going to summarize my various activities that can contribute towards efficient photodynamic therapy. Specifically in this talk, I'm going to present two of our, again, accidental discovery photosensitizers. The first one is hydrophobized, and the second one is photographic PS, which was developed at Kodak few decades ago. I'll also show you some of our molecular dynamic simulations work and some of the machine learning models we are trying to develop to study this photodynamic therapy. We recently worked with National Cancer Institute at NIH. They had this 𝗕𝗵𝗃 molecules that they wanted us to encapsulate in cholesterol or pegylated liposomes, which we were able to do successfully, and they showed very nice thermal, very nice PDT activity in mice. And since we like to ask why, I am also focused on combining experimental electrobinding studies, because hey, if the cell membranes are negative, I want to study if these photosensitizers are positively charged enough to bind with the membranes. And then how are they going to translocate? How are they going to interact with cell membrane or within the cell membrane? We study that using DSC. Once we have these experimental results, we feed them to our computational modeling simulations to actually visualize where these molecules might be going in. And then we are good to feed all of that data in a machine learning model. So I'm going to quickly talk about in just 3 or 4 slides, because I also want to focus on my other photosensitizer, which we thought was very exciting discovery. So 𝗵𝗃𝗀𝗃 is a positively charged, very tiny molecule that is well known to PDT community. But it's a very small molecule, so you can imagine how this tiny molecule will travel this bilayer. So we did a very simple experiment. We added a carbon chain or a tail to this molecule. So we added a carbon chain of 5 atoms and then 12 atoms. Simple. It's not as simple as it sounds, because purification is very challenging if you have longer tail molecules. And then we tested the cytotoxicity. And we learned that after 24 hours, because C12 has a longer chain, it inserts deep into the bilayer, it transports deeper. And so the uptake was much higher, and the cytotoxicity reported for C12 was much higher compared to C5, which was a small tail. Because amino is a positive charge molecule, and then our liposomes have slightly negative charge. This is not the best representation, but this is just the zeta potential of liposomes, which are negative. When we added these positively charged 𝗵𝗃𝗀𝗃, we saw the zeta potential was flipped to positive millivolts, which confirmed that there was definitely binding between 𝗵𝗃𝗀𝗃 and our liposomes. And we wanted to study how deep they go in the bilayer. So of course we used DSC, but in this work we used annealing. So we would add these 𝗵𝗃𝗀𝗃s to liposomes, and we subjected them to subsequent heating and cooling cycle. So if we have a small molecule with a small chain, it does change the phase separation a bit, but really between various heating cycles, you don't see much changes. However, when you have a long chain, what you see is phase separating domains. So in first cycle, while C12 was going in deeper, it created a lot of disturbance within the lipid bilayer. Now you have some DPPC rich domains, some DPPS rich domains, and some mixed domains. But with subsequent heating, C12 integrated well within the bilayer, and it brought back all the lipids together, and it kind of repacked them in the bilayer. So now you see a single nice peak. We published this paper in 2022 where we suggested that, hey, you can use DSE to predict changes in liposomal vaccines, such as COVID-19 vaccines, how they will change during the storage, transportation, and distribution, because during the storage, transportation, you might expect different temperatures of the environment. So DSE is a very quick test to see how things will change when there are temperature changes in the environment. Along with studying the length of the chain, we also looked at the concentration, because do remember that when we talk about concentration, we are increasing the number of molecules getting cooperated with lipids, which I talked about when I showed you the 𝗕𝗵𝘀𝗶𝗆 enthalpy equation. I'm not going to focus on that. I will focus on why we need to also study concentration, because that directly relates to the dosing. And the shorter molecule created a lot of phase separations here at high concentration. And that's because now those tiny molecules are getting integrated close to the head group, and they are aggregating, and then they are causing more phase separations. For C12 at very high concentration, we saw a flat line, and then we were not sure what was going on. So we did molecular dynamic simulations, and we learned that C12 disrupted our lipid bilayer. It kind of formed post that I showed you in the beginning. And so it turns out that this molecule can kill the cells even if there is no light or laser present during PDT. So we thought that was very interesting finding. In the recent times, we are also trying to learn machine learning, and our goal is to predict the phase transition, predict these phase transitions in lipid bilayer with respect to the chemical structures. So I'll show you some of our failed experiments with machine learning. So what we did, we used all the data from C5 and C12 I showed you in the previous slide to train our machine learning model following this architecture. And we tried predicting what's going to happen with C9. So C9 is somewhere between C5 and C12. So we thought, how hard would that be? So the blue line represents our machine learning model. The red line represents our experimental data. And we tested that at low concentration C9 and at different heating cycles. So we noticed our machine learning model was able to predict the location of the peak, where the melting peak would fall, but it did not predict these phase separating domains, unfortunately. And we also increased the concentration, and we saw something similar, that it was able to predict the location, but we also noticed that it kind of missed a peak at lower temperature, which could be just 𝗕𝗵𝘃. And so machine learning models are still in progress. We are open to collaboration. We have tons of experimental data that we would love to use to train the models. Just to quickly summarize this section of my talk, it's the combination of electrostatic and hydrophobic interactions that can drive the lipid bilayer interactions. We think the easiest way to do this is by increasing the length of alkyl chains that will keep getting inserted deeper into the bilayer. Through this study, we accidentally discovered that, hey, if you do thermal annealing on DSE, you can predict their thermal history and behavior. Again, I want to remind that synthesis and purification is a laborious process. And so if we have machine learning models that can predict the behavior of these alkyl chains of various lengths, it will save us a lot of time, because purification gets really challenging as you increase the length of the chain. How am I doing on time? Okay, I'll just quickly show you this very exciting work that we stumbled upon. I think I'll try to wrap this up in next 3 or 4 minutes, so we have some time to discuss. So 𝗕𝗵𝗀𝗀 is a lipophilic cation. That means it is hydrophobic and it's positively charged. It was developed by Kodak, and it was developed as a photographic emulsifier, and just as a dye or a pigment dye to make photographic films. We stumbled upon it and we wanted to see, hey, can I use that dye for cancer applications? And we looked at the electrostatic binding. It performed great. It bound. The negative zeta potential became positive. So we knew it was going to bind well. We tested the concentrations, and at all different concentrations, although it formed aggregates, it was still performing well in electrostatic binding. And then there was some in vitro studies done at our neighboring institution, and they confirmed that this molecule indeed has capability to form a reactive oxidative species that will kill the cells. And we did a quick DSC at different concentrations, different heating cycles, and we noticed that PS's, this type of photographic PS can also kill cells. It's very effective in PDT. And this work was done during lockdown, and so I was in between students, so we collaborated with University of Buffalo, and they helped us with computational modeling studies. This is a very interesting study, we thought, because they were able to predict where this molecule will go. So this is a lipid density map of both DPPS and DPPS. So the dark blue spots are DPPS. And we noticed this 𝗕𝗵𝗀𝗀 molecule prefers staying close to DPPC, which is this vitrinic molecule. So we thought that was very interesting. Then they also did some time series studies as to if you have a lipid bilayer of DPPC, DPPS all together, right? And then at what point 𝗕𝗵𝗀𝗀 will come in contact with the lipids and how well they insert in the bilayer. So I'm going to show you this mechanism through these videos. So the first mechanism that we'll look at is harpoon mechanism. And based on the time series study, we noticed that the first contact was made at 26.5 nanoseconds. And okay, so the video is not working for some reason, but that's okay. And then after 29.5 seconds, we kept running the simulations. And we noticed that the second contact was made at 134 nanoseconds. I wish I had the video to show this flip, but that's okay. And at the end of both of these mechanisms, the 𝗕𝗵𝗀𝗀 molecule had the same orientation. So here it was moving and tilting, but by the end of both mechanisms, it entered as harpoon. I wish the videos were working. Regardless, I just want to provide you a summary. So we do a lot of experimental studies on electrostatic binding, hydrophobic interactions using DSC. We combine those experimental studies and then we add them to our computational modeling studies. We are also trying to combine the computational modeling studies and experimental work in machine learning models, so we can apply all of that knowledge to various areas in which liposomes are being used, and we can save a lot of time before we enter our liposomes in vivo studies. So I have 5 more minutes. I just want to quickly remind you and leave you with open-ended questions that the interaction of therapeutics with cell membranes is a complex problem. And there's a lot of concepts of interfacial thermal and transport phenomena that is involved. And we can start by a simple technique such as DSC to see how things bind, how things change in lipid bilayer in terms of packaging and orientation. And of course, depending on the choice of lipids, their hydrophobic length chain, the saturation charge, this kind of phase change behavior changes drastically. And there are several applications where the thermal analysis of lipid and bilayer can be applied. One of the examples I showed you was photodynamic therapy, where we were able to notice the localized heating was can be used to design better carriers for photosensitizers. And I also showed you briefly how we can repurpose or rediscover the molecule. So if you know there's a photo sensitive molecule or a dye used in photography, we can use these tools to quickly vet out if they can be used for PDT applications or not. So with this, I'm going to wrap up this presentation. Thanks for listening, and I'm happy to answer any questions you may have. I think I have at least 3 more minutes.
Thank you for the really interesting talk. And if anyone has any questions, please feel free to ask Anju, over Zoom, or you can also type them in the chat.
Anju, I have to ask you. So when I met you, I believe in 2018, you were studying pool boiling, and I was really curious to know more about how you end up, working in this area, which arguably is very interesting one. Wanted to try to understand what was about, this transformation, and if someone else, for some reason, will decide to move to this area, what kind of step would they have to take?
Thanks, Matthew, for the question, and thank you for asking this, because I know in my first slide I showed you different interfacial thermal and transport problems that I'm working on. So actually it's the opposite. Some of this work is coming from my PhD dissertation. My PhD dissertation was about making self-penetrating peptides to kill some microbes and cancer cells. And then I moved to RIT, and I was trying to name my lab thermal analysis lab. And Satish said, my lab is called thermal analysis. I wish Satish was in the audience. And then we exchanged notes over coffee, what kind of thermal analysis you do versus what kind of thermal analysis I do. And somehow he pulled me into pool boiling, starting by saying, hey, can you help me with some graphene? And then one thing led to another, and I started working in boiling. So to answer your question, this was my training, my PhD training. I'm a trained chemical engineer, and this is what I was trained on. And then I switched to pool boiling, but I still continued working in lipid bilayer. And after moving to UT, we have an excellent medical campus. They do a lot of great work with cancer. And that's how I kind of took a U-turn to lipid bilayer. But then if you look at this problem, it has all the components of pool boiling. There is heat involved, there's phase transition. So really, I always feel if you can design one thing, you can design anything. If you know the concepts of, if you love heat transfer, you can solve a lot of problems in energy, environment, and medicine.
So what kind of suggestion would you give if someone is interested in, reorienting research from, more conventional pool boiling, fluid transfer to this research area?
So Matthew, I would say it just accidentally happened to me that I found a job in RIT and I moved, and I feel the easiest way is to collaborate with an expert. So I have been working on lipid bilayer since my PhD days, and the transition to pool boiling happened because I just happened to be in the same college as Satish, and we started talking about interesting things. And with my background in materials and love for chemistry, we were able to do a lot of other things. So I think the easiest way, if you want to make a transition or if you want to reinvent your research, is through collaboration. And that way you're bringing a different set of expertise to an established field like boiling.
Thank you. I think in the interest of time, I think 𝗍𝗵 has a question. So 𝗍𝗵, please feel free to ask the question.
Hi, Anju. Nice
Hi,
talk.
Thank you.
So I'm going to ask a question related to condensation, not really boiling. So I do some research work related to looking at how some of these monolayer coatings that we use to promote dropwise condensation, how they fail. And so some of your work with these alkyl chain lipids or molecules was interesting, because I'm trying to understand the limitations or the opportunities with your nano DSE technique, where you look at potentially how maybe I understood this incorrectly, but how these molecules change phases within the chain itself. Is that correct? Within the alkyl chain. And you were showing some of these peaks, and again, perhaps I misread the plots.
We can go back.
I was wondering how applicable this technique is to look at different types of alkyl chains, whether it's looking at some of these like thiol or silane molecules and trying to look at whether we can probe degradation of some of these other species or molecules with your DSE technique.
Thanks for the question, 𝗍𝗵. It's a very interesting discussion.
And if I'm way off base, please just say way off base.
Oh no, you're not. So just to start with a very simple answer, if you have a round molecule and if you add a tail, you will see differences in thermal behavior, because now the molecule will kind of rotate with heat and it will do its own things at molecular level. So you will definitely see some changes in nano DSE behavior. To answer your first question, you mentioned something about how easy. So nano DSE is a very sensitive technique. That means, so it has a very tiny slot to add your sample. And one disadvantage of nano DSE is you can only run solutions. So if you have solids, then you have to stick to DSE. But I think the recent DSE models are developed well to pick up any sensitive information. So nano DSE is only good for colloidal suspensions or solutions. If you change the length of chain of a molecule, you will definitely see some differences in their degradation behavior. For example, PEG molecule that I showed. So even if you ran a PEG molecule at different molecular weight, so this is one of the projects I give to my grad students in my graduate materials course, where they are given PEG molecules of different molecular weight. They have to run DSE, and we see shifts in the melting peaks and also glass transition. So definitely the length of the chain, the molecular weight impacts your DSE results.
Thank you, Anju. And perhaps I'll just send you an email and we can chat a little bit offline about potential collaboration opportunities, or maybe there's nothing to go forward with, but perhaps we'll just learn a little bit more. But I'll connect.
Sounds good. I look forward to it.
Thank you.
Thank you, 𝗍𝗵. I think probably in the interest of time, maybe I'll say that let's conclude today's session, and really thank Professor 𝗕𝗵𝘀𝗶 for the really interesting talk. And I think if anyone in the audience have more questions, please feel free to contact her via email. So again, really happy to see everyone, and we hope to see you again in our next session.